on velvet. And we can put on a petri dish. We can press the bacteria on. Then use another petri dish, another one, and another one. We'll look for different things. Do you know who invented replica print?
They should know.
They should know. Who? 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 All right.
Come on, guys. This is the homework I give to you first. Oh, yeah.
You sound like an owl.
Oh, eraser. Eraser. Let me find a clock.
Okay. Back in the early days of molecular biology, there were a couple of guys named Lurie and Delta. And they got the Nobel Prize for this fluctuation test. And it was just a simple experiment. Well, simple. So a cell's divided. Older version that when you hit a selection, that's when mutations occur in response. So if you stood on the ground long enough, you'd start to grow roots. Okay. Letter came from. And he invented replica printing. And what he did was if he grows E. coli and he plates down 10 to the 8 cells, and you wait a day, and now you get to, say, greater than 10 to the 10th as they grow on the LB plate. Okay? And then he replica printed this to another L plate. And then 3 more plates, for example. So the same original plate, you could replica print on the multiple plates. There's the L, L. These are L plus strep. So it's from the same original plate. He plated onto these 4 plates. And then he waited. And he got. Of course, this group, no, there's no selection. So he got the same pattern on each plate, right? So if mutations arose in response to the selection, it should have a random pattern every time. It didn't. And then you can go back to the original plate where you saw this colony and then find strep-resistant backs. Right? Showing that they were already in the population and you didn't need any fancy mathematics, right? Just a simple genetic experiment. Said the exact same thing. I just thought of that particular replica printing yesterday. All right. He's the guy. Lederberg got the Nobel Prize for discovering sex in bacteria. All right, we're going to talk about operon and gene. Is that right? Is that today's list? Yes, fusions. Okay, so the lab already had a little bit of an introduction, so you're going to get a refresher. Yesterday we mentioned the muphage and how that was used to make lac reporters. We cut out part of mu, right? We can put in lac. We put in its own ribosome binding site. And then an ampicillin resistance, or now we work with a smaller version. That's kanamycin resistance. Okay, so all you need for transposition is the ends. You have transposase, and we can put that next door to cis. So we talked about that yesterday. One. So when this mud lands in front of our gene, now since the lac operon in the mud has no promoter, its expression depends on this promoter, right, for X. So any positive, there's a positive or negative regulatory signals for X is now regulating lac. Lac is very easy to look at on indicator plates, X-gal, tetrazolium, conky. It's very easy to do beta-galactosidase assays. It's just converting a compound called ONPG. Cleave it and you get yellow color. Very easy enzymatic assay. Lactate is a very stable enzyme, so you just take the cells and you add a little chloroform to lyse the cells and do the assay. It's super easy. Whereas maybe your X protein might be very difficult to assay, right? This is very easy. So that's great. Okay. The other flavor we are working with in the lab, that Malcolm Casado invented. So these are transcriptional fusions. And then there's another one which has LAC-Z. No ribosome binding site. It starts at amino acid 7, so you don't need the very first few amino acids. And the original one, but 2, or the version we work with, the shorter version. mud K. So now, with mud K, since there's no ribosome binding site, you have to use the ribosome binding site for X to express lactate. But also, it has to be in frame. So the product of mud J, we got lac RBS, is part of X and then lac Z. For here, you get a fusion. You get that part of X that's now fused to lac Z. So this would be a gene fusion. Where translation of lac Z is dependent not only on transcription of the X promoter, but translation of the X gene. So you need to be not only in the right orientation, so that the promoter is expressing the lac operon, but also in the right reading frame. So with the mud J, it's a 50-50 chance to be in the right orientation, to be expressing from that promoter. Here it's a 1 out of 2 times 1 out of 3. So not only the right orientation, but also the right reading. So we should see that, I hope. If the gods are looking down on us, we'll see that today in the lab. Okay. All right. And I just wanted to actually give a little example of a time where my student was really depressed. And I don't understand why students get depressed. So there's two things that I think about genetics. One of the things is when you go in the morning to the lab and you look and you think, it's Christmas. It's Christmas every single day. The other thing,
Sometimes you've been a bad kid, though.
yeah.
Sometimes you get cold.
And the other thing my PhD advisor taught me was that every experiment works. No experiment fails. It may be that you screwed up, but inside that incubator is the truth. Right? It's the truth. Every experiment works. Maybe not the way you wanted it to work. Maybe you forgot something, but it's all a fun learning experience. Okay. So we talked a little bit about salmonella, swimming salmonella. And salmonella is a little different from E. coli. We mentioned this flagellum, lysine, right, that makes the propeller for the flagellum. Well, salmonella is different from E. coli in that it has two different flagellum antigens. Okay? And it can change between these two. They're functionally the same, but the surface amino acids are different. So that when your immune system makes antibodies to this guy, and he switches, it's like putting on a new invisible coat. You can be immune to antibodies against this guy. Okay? So it gives you some advantage. All right. This is called flagellar phase variation. And the lab of my postdoc, long before I got there, figured out how this works. And it works by flipping a promoter in the chromosome. Okay, so there's a promoter that transcribes, and I can just tell you, these names, fly, flash, blah. So that's just, if we look on the chromosome, remember I told you there's like 360 genes? Okay, so the alphabet has only 26 letters. Okay. So we name the flagellar genes based on where they map. So there's some that are here, and those are called FLG. Some that are here, and they're called FLH, FLI, FLG. So that's what they figured out, how to name these genes. All right. All right. So there's a promoter, and that promoter transcribes it all. This, we know what that is, right? A is an inhibitor of the other guy. And so A is an, we've got an inhibitor. So that when you express this operon, only the flag B flagellum is made, right? And in Mel Simon's lab, they showed that this promoter flips. Okay, so there's a recombinase called the HIN, because their old names were H2, H1, hapten, which is just an antigen name, hapten. So this is hapten inversion. And so when you invert that, what happens to this operon? It has no promoter. Great. So that means there's no repressor. And now we can make YC. Right? Flagellar phase duration. It was first seen in 1922, but wasn't figured out until 1977. Okay. But Bruce Docker first proposed that this was the repressor back in the 50s. So I simply asked my student, her name is Heather, to go ahead and imply C to make mud J, imply C, mud K. So we can look maybe for transcriptional regulators and translational regulators. All right. So she first wanted to characterize the repressor to show that the repressor was doing its job. Yes? All right. Draw it here. So we're going to look at beta-gal levels with a YC of J, and we're going to look at the YC, let's see, flag A on, flag A off. So Heather was able to make deletions. So these are invertible segments. So it recombines here and here. Combination sites. And if you delete one of them, they can't flip. So this is locked. Right, and this is locked. Because of the recombination sites, they're locked in one or two ranges. So, if we look at the mud J, and if they're there's no repressor, we get 100%. Okay? If we have the repressor, we get 20%. Good? Makes sense? What about if I see my K? Flash A on, flash A off. So there's no repressor. Again, 100% with the repressor, 0.5%. So this is what depressed her. She couldn't make sense out of this. I was so excited. It was just so beautiful to me. What did that tell us? That actually told us that this flag A is a translational regulator, not a transcriptional regulator. And sure enough, we're able to show that this makes mRNA and near the ribosome binding site, this flag A would bind the RNA. If we tested binding the DNA, it wouldn't bind the DNA. Right? So that was pretty cool. And we can look for mutants. So under the condition here, this is lac minus. What can we select for? Would be translational fusion. Cells are lac minus. What are we going to select? Lac plus. Yes. So under this condition, we select for lac plus, and we get mutations in this region where it doesn't bind the RNA. Nice. Okay. We also got a very cool mutation. So let's try this. Here's the ATG. And this is ribosome binding site. And it found, these are our mutations. Binds right near the ribosome binding site. Well, we got one mutation that actually had a duplication, where this part, let's see, this part here, actually even in the coding, got duplicated. Duplication. So we have a ribosome binding site and another ribosome binding site, ATG. So this was lac plus. Yeah, in the presence of the translation inhibitor, but without the flag A, with the flag A off. Flag A off. Lac minus. So we selected for lac plus in the presence of the inhibitor, and that's what we got. But this particular mutant made this duplication that now turned off. And what that meant was this inhibitor had to bind here to prevent RNA folding, so that now you can translate from this ribosome binding site. So we changed it from an inhibitor to an activator. Anyway, so these are things you can find out by working with not only the transcriptional fusions, but also the translational fusions. That was a fun one. And again, you should never be depressed by getting weird results. Right? They're always telling you something. All right, so I'm just going to tell you just variation on a theme. So mud is great. Lac is a great reporter. And Sam and I both taught this course. Cold Spring Harbor. Here's this donor that we used. There was a guy in the class that thought this was really cool. So he went home to Belgium and he wanted to make fluorescent fusions. So he just cut this thing out. Replace the kanamycin with chlorophyll resistance. And this is yellow fluorescent protein. So you can use that to make fluorescent protein fusions for things that are Ian in my lab did a similar thing where she cut that out. To make beta-lactamase fusions. So beta-lactamase, you know what that is. So this confers ampicillin resistance, right? But only if it's secreted in the paraplasm. Okay? So beta-lactams work on cell wall synthesis. Right? And beta-lactamase normally has a secretion signal. Right? And that secretion signal takes it to the secretion system, and it's a cleavable secretion signal. And it puts the blah in the paraplasm. There's an enzyme in there called DSPA, disulfide bond forming enzyme. So blah actually has to form a disulfide bond. So in the paraplasm, it's reductive. So there's no, the disulfide bond won't form in the paraplasm. It has to get here and won't form in the cytoplasm. It has to be secreted, and this enzyme can now form the disulfide bonds, and this confers AMP resistance. Okay? So this, if you can make fusions, if your X gene, for example, has a secretion signal in the N-terminus, you can find it. So you can do a mud blah hop and look for AMP resistance. And that will land you into a gene that has a secretion signal to put it in the paraplasm. Okay? So for us, we did an experiment that's related to Professor Antonio's work. Remember SPY 1? So we have a CPOT driving hill A. Right? So this is test turning on SPY 1. Yeah? And then we can do a mud blah hop, which is chlorophyll resistant, into the strain and look for TET, AMP resistance. Okay. So finding SPY1 genes whose proteins have a secretion signal that take it to the injectosome. Now we cheat a little bit. We don't have the needle, we just have the secretion apparatus. So we delete the Mg secretin, so that's gone. So anything that gets secreted is going to go into the periplasm. And we get some interesting things. One of the things we got, so back to regulation of SPY1, we have delay, well, turning on the injectosome gene. And remember, you have normally it was this, and you got this secretin, and you got a needle, and then you put on a translocon. And these are CYP, E, C, E. And before the translocon goes out, you just have a needle. And you have the CYP, E, C, D. They are protected by the SIG-A, which is the type 3 secretion chaperone that protects these in the cytoplasm and delivers them for secretion. And then once they're secreted, SIG-A plus MF turn on effectors. Yes? Okay, so that's a way of not, it's like in the flageller, you don't make the propeller until the motor's built. You don't make the effectors until the needle is built. Okay, pretty cool. Well, we found that one of the guys we got was in fact SIG-A. We were not expecting that. But that's genetics, right? Can't always get what's wrong. Remember that? And so this is nothing we published, but what we think is going on with SPY1. It's analogous to the flagellar system. So in the flagellar system, we have this flag M, we talked about binding sigma 28, and now when this is complete, he goes out, and now we can turn on YC, right? We talked about that yesterday. But this result, you smoke little funny stuff. You can imagine that this is really the same thing. So kind of a convergent evolution. Turns on included in those genes SIG-A. Right? And so you can imagine early on, what if SIG-A is now being secreted during the growth of this structure, right? So we secrete SIG-A, so there's no activator, and so here we have an inhibitor until you're finished, and here we get rid of the activator until we're finished. And now we keep it inside to interact with BCD, and then these go out. Now we turn on late. So an inhibitor goes out after completion. Here we have an activator going out before completion. But it's the same way of doing, different way of doing the same thing. All right.
Is it possible that it's going out also through the type 2 system?
It's depended on the ejectocell type 3 secretion.
Yeah.
Yeah.
But I mean, once it's formed, but the possibility that maybe it's not going to the peripheral.
When you have the
So they
switch,
show that. But maybe it's because you force a little bit because you have an injury.
when you have the switch, when the needle's complete, then it stays inside. And it can interact with BCD to get dimmer.
So we can test that, the last portion, because we have the CK tag with a different, at least 2 tags. We
Yeah.
can check that it's not being secreted.
Right. Yeah, we haven't used the blocks. We can just look at beta lactams as well.
Yeah.
Anyway, so that's just a fun way of using fusions. I think Stanley's going to tell you about some more OA, right?
I thought you were going to, yeah, I'll talk about it.
But the point is, you can make whatever you want to make, right? You can make fusions. Whatever you want. So they're very powerful.
It's got drug-resistant marker. And it has part of the back operon. And then OA is alkaline phosphatase. In the cell, alkaline phosphatase, you don't want to turn on inside the cell, right? Because you have too many really important phosphorylated compounds. For example, nucleic acids, nucleotides, and so on. Right? So just like Kelly described with ampicillin, alkaline phosphatase, the OA gene product, is exported into the periplasm. And in the periplasm, much like he described for ampicillin, OA can form disulfide bonds where it's a dimer, and it's the dimer that's active. Okay? These dimers can't form inside the cytoplasm because the reducing environment is such that they can't form. In the case that he described with beta-lactamase, it's intramolecular disulfide bonds. In this case, it's intermolecular disulfide bonds. Right? Very, very similar thing. In the process of doing this, we insert this into a gene. If that gene has a promoter and has the appropriate signal sequences for export, what you end up with is you end up with a little hybrid protein that has part of this beginning attached to a lot of OA. Okay? So, very simple, very similar, simple approach that can be used to look for a lot of different things. This will work in some cases where you may not be able to use beta-lactamase because of inherent resistance or whatever in your organism. Right? This was developed initially by a guy who's a colleague of Kelly's back when he was at the University of Washington. But one of the cases that I know of where it was used involves a case where you're thinking about Vibrio cholera, right? Right? So with Vibrio cholera, we knew that cholera toxin was made for a long time. And there was a lot of evidence that that cholera toxin is what is responsible for the disease with cholera. But people didn't understand how cholera toxin was regulated are how it was made, right? And so one of my colleagues, Mon Taylor, when he entered his postdoc, he said, well, I'm going to try to find this out. And so they looked and looked and looked for mutants, and when they looked for mutants, the big thing that they found were cholera toxin mutants. They had a hard time finding anything else that affected cholera toxin production, right? But so then he went back and he said, okay, well, I'm going to try to look, I'm going to just look for alkaline phosphatase positive insertions. When you look for alkaline phosphatase positive insertions, you'll find everything in the cell that is secreted, everything, right? But there's many, many fewer things that are secreted than there are total proteins made by the cell. So he's narrowed down the number of guys that they have to look for. They then looked for those mutants that affect cholera toxin production, and in that case, in the first time he did it, he found one mutant that was blue and didn't make cholera toxin, right? The world looked and looked and looked at these things, but because they had not narrowed it down enough, it just got lost in the wash. They just couldn't find it because the test for this wasn't strong enough. In vitro test wasn't strong enough at that time. Now, yes, it's a different story there. But so this is another case where you can do that really effectively. The take-home point of me talking about this, when you've already heard about beta-lactamase, is that some of you work on different organisms, right? So we say, oh, we use kanamycin, we use ampicillin, we use tetracycline, we use all these things in salmonella. In some organisms, certain antibiotics don't work, right? In some organisms, you may have a situation where the organism is inherently carrying multiple copies of beta-lactamases, or multiple ways of producing ampicillin resistance. So you may not be able to use that one, but you might be able to use this approach. Or you may have something that has real strong alkaline phosphatase activity, and this one wouldn't work for you, and you may be able to use beta-lactamase. Or neither of these work, and you clone your favorite reporter in between those guys and use it in your organism, right? So the real important point here is that we're giving examples. Those examples work really nicely in salmonella. In your favorite organism, you may have to tweak things to get them to work well, right? But it's worth doing because of what it will tell you. Huh?
Can I say one thing for the coffee break?
No, you can't.
Sorry, I just realized. Gene. The mud is at, after base pair 500. That you have 500 if the mRNA is not translated.
Hey, you want to have one other story before we go on break? So Kelly told you about the Poisson distribution, right? And many students suffer through the Poisson distribution. Poisson was a French mathematician, and he developed this approach because he wanted to understand the probability of success in gambling. He was trying to prove that by gambling more, it doesn't necessarily help you succeed more, right? But it was a very esoteric approach understood by mathematicians, very few people understood it. But soon after he developed it, the Prussian army had a big problem. And the problem was that especially the senior officers in the military, which used horses at that time, senior officers in the cavalry, some of them every year got kicked in the head and killed. Right? And now, the French soldiers probably didn't mind that, but it really influenced people. Got so they were suspicious and they did not want to become an officer in the cavalry. So it was really influencing the Prussian military. Right? So they hired a mathematician to come in and analyze this process, because they thought, well, maybe the officers get sloppy, or maybe the non-officers are training horses to kick officers. And so they applied the Poisson distribution, and it showed that, in fact, this was a random distribution. They accumulated this on cavalry across Prussia, and it fit perfectly the Poisson distribution. And it was the study on horses kicking and killing humans in the Prussian army that made the world appreciate the Poisson distribution. Let's have some coffee.
Algunos avisos, say that in Spanish. Well, in English. Okay, for you guys. Good. So, as you have seen, there's a photographer going around, Martin, over there. So, if you guys want to take pictures with them, it's a good opportunity to do that. So, eventually, we're going to make all the photos for free. We're going to post them on a drive, so they're going to be for free for all the people here. The other one is when the theory class is done, around 12:30, we're going to get the group pictures. So we are not going to follow what I wrote originally because we have school too close. So anyway, the other announcement is the guy at the copy shop, Señor Gabriel, the one in the copier machine. He says that a few of the manuals, it seems that they have a problem or they're not complete or there were some mistakes. So you guys can go and he'll fix it for free. So because he did it wrong, you just check them. Whoever who has them, just go and check and he'll do it for free. And if you guys still want it, you can still buy it from them. They're open today because we have the ETS exams. So they're making copies for everybody. And then the other thing is that manual you sent me this morning for everybody. Can I make it available for everybody?
Sure.
Sure? We can charge them.
No.
Yo no quería.
It's electronic.
Yeah? So, Stanley sent me this morning, a manual. He has like printout or something like that. So, I think that was already published online for a while. You had a website, didn't you? Salmonella.org?
Yeah, but I didn't publish that one.
Not that one. Anyway. So anyway, I can share it for you, so I'm going to put it in the drive. I'm going to make a special folder, or maybe just put it in one of the either discussion or reference papers in one of those. Okay? Okay.
So it has protocols for how you can do chemical mutagenesis when you need to. Also, some different versions of how you can use transposons on things and stuff like that. So it does not supplant the protocols that those of you in the lab are doing. If you're not in the lab, you can have those protocols, and it expands it because it was a longer course.
Sí, ¿verdad?
Vamos a continuar, ¿sale? Para terminar a tiempo. Yours.
So we're going to do something a little bit different now. We've been talking about reporters that you put into your organism and you look for particular genes. Now we're going to tell you a system where you can take DNA and proteins from your organism and test it here. So this test is about how regulation happens. How a repressor binds to a site, right? Or it could be how a nucleic acid binds and represses. And how can we get mutations that change that binding in ways that help us understand the regulation and in ways that allow us to do new things. Very different system, all right? The whole system relies on everything you know about the lysis-lysogeny decision of bacteriophage lambda. Everybody's heard of bacteriophage lambda, see? Okay. All right, so you remember in bacteriophage lambda that there's this set of genes, some of them are involved in producing new phage particles, but a series of them are important for the regulation whether lambda becomes a lysogen and integrates into the chromosome, or whether it lyses the cell and makes new phage, right? And the whole thing with bacteriophage lambda is dependent upon how much of the C1 repressor is made. Right? Pro is a key signaler in terms of telling how much C1 is made. Okay? A beautiful, one of the most beautiful books I've ever read about genetic regulation is this book by Mark Patashny. It's a little bit old now. Beauty lives forever. So if you can read this book, I really recommend it. A Genetic Switch. He has newer versions, but A Genetic Switch is really a great book to help you understand this. There is also, by the way, Mark Patashny is the one who first purified this repressor and showed how it worked, right? And there's a YouTube that you can watch that Professor Antonio will give you the link to, called the Race for the Repressor. Because Mark Patashny was working very hard to isolate the lambda repressor. At the same time, at the same university, another professor was working really hard to isolate the lambda repressor, the lac repressor. The lac repressor. Okay? So it was a race. They both, the first person to isolate and characterize the repressor was going to be famous. Very interesting little video that tells about this and gives pictures of people in the lab, including some undergraduates in the lab that ended up being famous. Good to watch. Okay, so this is lambda. You know about it. Bacteriophage P22, most of it is exactly like lambda, except they changed the names just to make it harder. Okay? So this part of bacteriophage P22 is just like all of this stuff in lambda, but instead of this C1 protein, it's called C2. C2 is virtually identical to C1 in lambda. Okay? So this region here is called the immunity C region, for control. As you know, from lambda, when the C2 gene, when the repressor is made, that repressor has to multimerize. And when the repressor multimerizes, it comes back and binds to these sites and turns off lysis. So you become a lysogen. Right? So, if it binds, then you get lysogeny. If it does not bind, then you get lysis. Protein DNA site interaction. site, right? It relates to everything you've ever learned about proteins binding to DNA. Now, this is how lambda gets regulated. P22 has a second region called immunity I. Right? And immunity I is made up of a gene called MNT, and there's a promoter called pAnt that expresses the ant gene. Ant gene is anti-repressor. When the ant gene is made, ant gene binds to this and prevents it from binding DNA. Right? It makes sense. Anti-repressor. Right? Now, this gene is called maintenance. This gene makes a protein that binds here and turns off ant. So now if you're not making ant, whether you form a lysogen or not is determined by this process, just like in lambda. In biology, so many different ways of doing the same thing, right? Okay. Now, this was really thoroughly studied. Bacteriophage P22, lysosligogeny. People have done a lot of work on this. And a friend of Kelly and mine, called Phil Yadarian, got a great idea. And that idea was to take this P22 system and build something called the challenge phage that would allow you to study DNA protein interactions. Okay? So specifically, how a protein binds to a regulatory site. Okay? Phil Yadarian is one of the most unusual people I have ever met in my life. Well, Kelly, tell a story about Phil Yadarian.
Really?
Yeah.
Really?
It's important to know that there are a lot of diverse people who are successful scientists.
I can tell you. So, I did my postdoc working on this system. And I was gonna study this. Remember I talked about that flipping promoter? So in this system I put the recombinase binding site, the part that the recombination occurs. And I can look at the protein that does the recombination, but just as a repressor. So I can just look at DNA binding independent of recombination. Okay? So I did it because I saw Phil Yadarian give a talk about this and I thought, wow, that's incredible, right? So the first 6 weeks of my postdoc, I went to Phil's lab to set this all up. And every day, Phil would take me to the liquor store so you could buy a case of beer and a bottle of whiskey. And every day, he'd drink that case of beer and that bottle of whiskey because he was trying to stop smoking. So every time he wanted a cigarette, he'd pour himself a little shot of whiskey. He didn't have to have the cigarette. And then the other funny story, just years later, I think Stanley is alluding to, it was a Sunday at 2 o'clock in the morning, and I get a phone call. I answer the phone. Kelly, it's Bill. I just want you to know I really love you. Right, Bill, I love you too. Anyway, he's a character, okay? But he was also really, really smart, right? And he made a very nuanced system that it's really a tricky way of going. So the whole idea here
side, you could control the lysis-lysogeny decision, right? You can select for lysis by isolating phage, okay? And you can select for lysogeny by isolating an insertion of this that carries drug resistance. Okay? So this gives us a set, you'll see in a minute how that works, but gives us a very strong positive reaction for changes here that either prevent protein binding, right? Or changes elsewhere that allow protein binding. Both ways. Let me show you. So he set this up so he has a derivative like this, but instead of the normal PN, it's set up so you can easily replace it with your favorite sequence. Your favorite DNA binding site. Okay? So, this has been done with a lot of different things. Kelly's used it on this Hen-Hicks inversion system. We've used it on Lysar repressors. Other people have used it to understand restriction enzyme binding. A whole bunch of different things. But you can insert very easily your favorite sequence here, right? And then you take the protein, your binding protein, DNA binding protein, and you express it under control of the CAC promoter, right? So in this case, you can turn it on at high levels by adding IPTG. Okay? So this is set up. It's got your protein here, your binding site here. Okay? So if you put your binding site and there's no protein that can bind to it, this is going to always grow as a phage. It'll always make ant. It will always inhibit the repressor. It will always form phage. Easy to get the phage. You take a lot of bacteria on a plate, you dilute the samples, and you can isolate phage from the plaques. Now, if binding happens, now you're going to get a lysogen. That lysogen will form a colony, and you can isolate that colony. I should show this. So you can isolate a colony by drug resistance. Right? So you either select for the phage when you want to prevent binding, or you select for the lysogen when you want to have binding. Right? Now, let's just say that you start out with your favorite sequence and your favorite DNA binding protein. Somebody give me a DNA binding protein and a sequence. Tell me.
CRP or Lexane.
Oh, okay. Let's just say this DNA binding protein is CRP.
Or cap.
Heck,
You like the cap.
Nah, I don't want
Either
to.
way. Well, sometimes you say crap. Students don't like
All right. So you bring in the CRP binding site, you put it on this phage. That can be done easily by recombination. And then you're expressing CRP off of this PCAP promoter. So if you put this site here and there's no IPTG, in that case you're going to get lysis. If you add, if you express CRP, CRP will come bind to this site and you'll get a lysogen. So good. Now, let's say we want to know which bases in the CRP binding site are important. So now we mutate. We have this repressed cell that's forming lysogens. We treat the phage with a mutagen. Sometimes we'll get a mutation in this site. Now it will grow as a as a living phage. It can never be repressed by CRP because CRP cannot bind to that site anymore. So we've identified an important base that prevents the protein-DNA interaction. But now, if you want to understand the interaction, what we'd love to do is we'd like to isolate a mutant of CRP that can bind to the mutant site, right? So now that mutation has changed the binding site, so it recognizes the mutant site. So now we mutagenize this plasmid, we get a mutation in CRP that alters its binding specificity. And now the CRP can come bind to this mutant site, and what happens? Okay. And the lysogen that we get, we can select for by requiring kanamycin resistance. Right? Now, a mutation in a DNA binding protein that recognizes a mutant site often is extremely rare, right? It's a very hard thing to find. It's hard to study. But when we grow these phage, we can get 10 to the 9th phage easily. We can find rare events on the phage. It's really easy for us to look for a kanamycin-resistant colony as a lysogen. So we can find something in the order of 1 in a billion. Right? Rare events. Okay. Once we find this guy that binds, we can now say, okay, let's find out what this did, how it changed binding. So now we can mutate this site again, so it doesn't bind, and we go back and then we isolate another one that binds. So you can continue this cycle to understand every base in that DNA binding site that matters. And not every base in the region matters, right? But the ones that matter will give us these mutants. And we can understand precisely the amino acids in that protein that recognize the DNA and how they do it. We're not making guesses from a structure. We are getting mutants that tell us those interactions happen. Pretty cool, huh? Right? So this is really a powerful thing. And the point of this is you can take something out of Tsugamushi and put it in here, and it can work in this system. As long as you can express your protein, and as long as you have an idea what the binding site is, you can do it. Okay? You have to be able to plaque phage, and you have to be able to replica plate. Right? So the tools in the lab really apply directly to this. Right? Now, this can be done in a number of different ways. So one of the papers, a paper by Fisher et al., is one I suggested to you guys to take a look at, because I think it really emphasizes something very important. Right? This is kind of splotchy. Let's just change this. All right. So restriction endonucleases, we all use all the time, right? They function by binding to specific DNA and cutting the DNA, right? And sometimes it would be nice to have a little more variation in the kind of restriction enzymes we could get, right? It'd be nice if we had more that bound to different sites, right? And so a couple of colleagues of mine, when I was at the University of Illinois, said, we had been using this system in my lab, and they said, let's try this with restriction enzymes. So they put a restriction enzyme onto the plasmid, and they put the restriction enzyme binding site onto the phage. Okay? Now, if you express the restriction enzyme, what happens?
It's going to cut
It will cut if it has its cutting binding site on the phage. Right? So what you do is you do this with a part of the binding site that doesn't allow the cleavage site. Okay? So now what they do is they modify that site, and this can't bind, so now you're going to get only lysis. Okay? They isolate a mutation in the restriction internuclease that changes the site so that it can now bind to that mutant site. Now, when you take this restriction enzyme and put it into a new cell, it has a distinct and different site specificity. Right? You can run this through many times on a single restriction enzyme. You can get minor variations in that site. So now you've expanded the repertoire of things that you can use that restriction enzyme for. Right?
A question.
Yeah.
How do you put that sequence there? If you're not, yeah.
How do you put this sequence?
Your YSS, your favorite sequence. How do I put my sequence?
Right. So it's easy to do that by a recombination, a simple recombination. Down here you clone it in. It's easy to clone on the plasmids. Here it's a recombination thing. Okay, so it works with repressors. It works with restriction enzyme, right? Kelly did this with an invertase. That's a recombination enzyme. It worked for that. People in my lab did it with lyse-R regulator proteins. Other people who have done this with nitrogenase DNA binding proteins from rhizobium. People have done this with specific DNA protein interactions from Klebsiella. Works in a whole bunch of different things. The other thing that you can do sometimes, though, is not all regulation is by protein-protein interactions. Sometimes it's by protein-nucleic acid interactions. So you can, instead of sticking a protein here, you can stick a different nucleic acid that you're expressing. And now this nucleic acid interacts with the site and can prevent it too. So that works as well. Pretty cool, huh? It's pretty sophisticated, but again, the power of a positive selection. We have a positive selection for binding, we have a positive selection against binding. So we can play this game over and over again until we understand at the atomic level all of the interactions that go into that regulation. You want to add something?
Yeah.
Yeah.
Yeah. Just show you how powerful this is. In the case of what I did, there's this recombination sites, and they're called Hicks. And so I was looking at the N recombinase, where we put the Hicks sites here, right? And my protein is N, that I express from PTAP. And so, like Stan said, if ANT is on, you're lytic, right? But if ANT is off, you're lysogen, which means you can become canamycin resistance. Okay? And I had a colleague in the lab who was doing the DNA work, and what Anna Glasgow showed was there are sites here which were major groove binding defective and minor groove. Okay, so there was a major groove and a minor groove that you could, this is a methylation interference assay in vitro, but she showed that there were these two sites. Time to go?
Oh.
Okay. So, point is, if we express this, we can get 10 to the 8 cells plus 10 to the 9th phage. Excess phage stimulates lysogen. And we get 20% of the cell become hand resistant. Right? Again, we're working at a scale of 10 to the 8. If you mutate in the major groove binding site, the frequency goes down to 10 to the minus 5. Okay? That's the major groove. If you mutate the minor groove, it goes down to less than 10 to the 8. So using this in vivo, you just say, okay, it binds major and minor groove, but we can now say that it actually binds primarily through minor groove interactions. So it's the first protein that was shown to be primarily binding DNA through the minor groove. And this challenge phase allowed that to see it because we have 8 orders of magnitude in terms of frequency of lysogeny that we can look at. That's really powerful.
And you reminded me to say something else. So we've talked about finding mutants in these sites, but you can also just change affinity for the site. Right? So that's an important thing. When you turn on this derivative, so here we're looking at phage. And this is the concentration of IPTG. Right? And by the way, you don't have to use PTAC. You can put your favorite promoter here. This is just a plasmid. Right? So the amount of IPTG you have. So if you have very little IPTG, you're not going to be making much protein. You'll get lysis, right? So you have a lot of lysis, and the more IPTG you add, the more lysogens you get. Okay? So what this means is that you can add very little IPTG, so you're making very little of your protein, and find the rare guys that binds to the wild type site better. So you're increasing affinity of your protein for the wild type site. So you can do that as well. And that's just by making less of this protein. You know, you turn things on with IPTG, boom, you've got a ton of those. So you just turn that real stat down so you're making much less protein, and it's an affinity change. So, and that will allow you to if you have a protein that's really important for some particular process, sometimes that affinity can really be high. Again, the restriction enzymes are an example where they develop new kinds of restriction enzymes using this approach, and those enzymes are used in any difference. Okay, let's see. We have talked in terms of reporters. We have talked about reporters that allow you to identify gene products that are secreted from the cell, to allow you to identify gene products that are produced under specific conditions within the cell, and to identify protein nucleic acid interactions specifically, right? So these kinds of reporters allow you to do a wide variety of different things. And if they don't do exactly what you want, it's a toolkit, right? That toolkit doesn't mean that you have to find the exact tool you need. It means knowing that this tool works well, you can build a better hammer for your particular use. And that, my friends, is advanced bacterial genetics. All right. You want to talk about the paper?
Sure. So, I'm going to talk about a couple of papers. There's the one about the Burkle dairy, we'll talk second, and first we'll talk about this one related to Hill D, since we all know and love Spy 1. And I'm going to start. So remember, flagellar gene regulatory system, there's this master operon activator class 1. And in 1999, my competitor, Kazuhiro Kutsukaki, he published that this gene, the promoter for this operon, actually had 6 promoters. We call them P5, 4, 3, 2, 1, and then there's 6 in here, 6. Okay, so he published that there were 6 promoters. Ah, that's a lot of promoters for one gene. So I had a postdoc in the lab named by the name of Pete Wooten, and he used a class 2 mud-lac fusion, J, and he could knock out all of these promoters except one. So you can have P1 only, P2 only, P3 only, P4, P5, P6. Right? What's important? So if we have wild type, we get 100%. If we have P1 only, we get 50%. P2 only, less than 1, less than 1, less than 1, 50%, less than 1. Okay. So that told us that probably the way he looked at these promoters was just looking at primer extension from the transcript. And so it's probable that these guys were just breakdown products as that RNA is being degraded. That's what we concluded. So it suggested that really only 2 promoters, right? P1 and P5. Then, all right, so let's get rid of those guys. And then with this class 2 mud, we do a T-pop pod. And we look for T-pop that was TET, TET-lac plus, TET-lac minus, and this was on this tetrazolium indicator plate that we're working with in the lab. And this wouldn't have worked if we used any other lactose indicator plate. So that's called luck. Okay. So it turned out that that T-pop landed in front of the HIL-D. And it was between the HIL-D gene and its normal promoter. So now you don't have HIL-D made. Okay? But you can turn it on by adding TAP. So it turned out then what happened was that if we knock out, if we have P1 only, we look at motility, these guys are motile. And these guys are not motile. Okay. And we saw this, it looked like HIL-D was regulating, and if we measure without TET, and we look at the class 2 lac, wild type being 100%, this was 50%. Okay. And if we look at P1 only, right, we saw no effect on, we saw actually, if we put P1 only, how do I do this? Sorry. No, I gotta remember. P1 only versus P5 only. Without HIL-D, that's 1%. So without TEP. So that suggested that the HIL-D was was actually turning on the P5 promoter. And that led us to look at that. And so, what happens, and you saw from the paper, that in fact, LD really does bind here as an activator, right? We showed that in the paper. So the class 2 has the hook basabite genes, and it has this sigma factor gene. It's in an operon with Z, and Z activates HIL-D, right? And HIL-D, also in that T-top experiment I talked about yesterday, one of the repressors was something called RTSB. HIL-D actually activates the operon. Well, HIL-D turns on anyway, but there's an RTS-A and B, and RTSB actually inhibits this, whereas HIL-D then activates this. So when we turn on the cells, we first have to turn on P1 to turn on Z, to activate D, to then turn on 5, to turn on B, to turn off 1. Right? So there's something weird about salmonella, which I guess maybe Olivia has some way of explaining this. Why is it as the cells are going through their growth phase, they want to make flagella, but then now they want to put it under control of spike line. Right? So that's what that paper is about. If you have specific questions about any assays, we can talk about that. But that's the story, right? So I'm going to now
I have a technical question for you
Yeah.
about the. So, in the old days, we used to do
when I was young.
in those days, we used to do footprintings with radioactivity. I still work with them.
So in that
So
paper,
now-
we have the HIL-D footprint.
Yeah, I know.
Right,
I know, it's
okay.
beautiful. And you guys did it with dioxycycline.
Hanna Singer did this, yes.
Yeah. So that was more a technical question. How, because if gels are like this size,
Yeah.
you just gotta cover everything with the solution and immediately take it either to a film, like we used to do it with radioactivity.
Yeah.
And now you don't use that, but it's still,
No.
the
Still pretty.
precision is really pretty.
Yeah.
I'm just surprised.
Oh, okay. you don't not believe it.
No, I do believe it. It's beautiful. But it's just
Well,
very
so
curious because we would love to do something like that.
the people responsible for the work, it's the students that make it beautiful. Not
Yeah, I know.
right?
That was when I was a student.
Okay.
I know that.
If you have good students, you get beautiful data. And those are my German students, so that's why it's so beautiful. They're very perfectionists. So let's talk about Berkeley area. All right. So why should I care about Berkeley area? Well, I care about everything. So, I have there is a professor in the medical school. His name is Ted Luke. And he works on cystic fibrosis. So he's the head, he's a pulmonary physician, doctor. We're doctors, but he's a physician out there. And he's in charge of the Adult Cystic Fibrosis Center at the University of Utah. So you probably know that a lot of CF patients, their lungs get full of bacteria, and a particularly important bacteria is Pseudomonas. Pseudomonas aeruginosa. Yes? Yes. And so what Ted had noticed is whenever his patients got a Burkholderia infection, so Burkholderia is pretty lethal to cystic fibrosis patients. If you have Pseudomonas, they can start treating it, keep you alive, but Burkholderia is much more lethal when it gets into their lungs. In fact, it was sad because in the early days, they wanted these CF kids to have people they can relate to. So they would have picnics with fellow patients. But if any one of those patients had Burkholderia, it would give it to all the other kids. So they had to stop doing that. Right? Anyway, so what Ted had noticed was that when his patient got Burkholderia, very quickly the Pseudomonas would go away. And he thought, maybe the Burkholderia is producing some antibiotic that's inhibiting the Pseudomonas. Okay? And he was working with a mathematician who's actually in my department, and that mathematician had said, oh, you should go talk to Kelly Hughes. Maybe he can figure out some genetic way of investigating whether this Burkholderia is producing an antibiotic. But the Burkholderia that makes people sick is called cepacia. Is that right? That's right. I'm sorry, I don't want to work with something that's going to make people sick. So I decided to see if this works. I took a Burkholderia species that was not a BSL2. Okay, so we decided that we'll start with one called thailandensis. Okay, that's something we could work in the lab, and I wasn't afraid that someone might catch it or anything like that. Okay. So that's why we chose that organism. And so I had a friend, actually, Stanley mentioned this colleague I had in Seattle, and his name is Colin Manoyle. And Colin was already studying genetics in Burkholderia and Pseudomonas. And what Colin had already established in E. coli was a suicide plasmid delivery system for transposons into Burkholderia. So there is an F-like plasmid, a suicide plasmid, I don't know, R100, I'm not going to try to guess, I can't remember. But on that plasmid was a transposon, T23 he called it. And this was missing a replication. This is kind of a famous delta pair, which is an important plasmid replication protein, which they express and complement in the chromosome, so that when this E. coli replicates, the plasmid replicates. Yeah? But this is a conjugatable plasmid, and if you put it with Burkholderia thailandensis, this can transfer that DNA with the transposon, T23. And that T23, he designed it such that it had trimethoprim resistance. And our thailandensis was fortunately sensitive to it was AMP resistant, it was resistant to all kinds of stuff. But it was still sensitive to trimethoprim. And also, he had a promoter that was going out, which was controlled by rhamnose. Okay? Rhamnose-inducible promoter. But what we found out was this might be a rhamnose-inducible promoter in E. coli, but it's not a rhamnose-inducible promoter in Burkholderia. So the thought was that if we introduce this onto Burkholderia thailandensis, and we have a lawn that's full of salmonella, and we put in there the Burkholderia colonies selecting for trimethylperm resistance, we could get some guys that had a zone of killing. Okay, so we got insertions that had a zone of killing, and the idea was either you have a repressor of the antibiotic gene or an activator of the antibiotic gene. And if you have a T23 in the repressor, they're going to be antibiotic on. And if you can get it in front of the activator, you could have maybe a rhamnose promoter that turns on the antibiotic, right? So that was the idea behind this transposon. But again, this didn't work. So it'd be nice to go back and fix this so that we have an inducible promoter that works in Burkle's area. Okay. But it turned out, though, that we could make insertions in a repressor. Okay. And I think that's called the ATSR. Try to remember. See? And so these were insertions in a gene called ATSR. Okay. So if you knock out that ATSR, you're now producing the antibody. So if we do that, we've got a nice, every cell. If you have this guy, then every cell is clearing the salmonella. We have an antibiotic. Yay! What is it? And so what Chris did, this is Chris Wozniak, using a technique that we will talk about tomorrow, was in the Burkle's area, he was able to delete ATSR. Okay. So now they're all constitutively producing the antibody. All right. So now with this mutant, we can go back and bring in the TN23, or the T23, whatever you call it, and allow that to hop by selecting trimethylperm resistance again. And now look for mutants in the delta ATSR. Mostly look like that. Right? But now look for guys that are not producing antibodies. Alright. And you got things in a bunch of different places in the chromosome. I got that. I don't want to belabor the ones that we couldn't figure out. Where is he? Right. So that was pretty cool. So we can use genetics, right, to find new antibodies. And he found this section of the chromosome with all these genes that produced a compound that had already been known called Thailand. So when you got insertions in the Thailand synthesis genes, they stopped producing the antibody. Okay? And this Burkle's had been known to produce other antibiotics, right? And that's in the paper. And he was able to show that if you knock out those other antibiotics, they didn't affect this. So this was specific to the salmonella killing was specific to Burkle's and the Thailand. And he characterized that in Eric Schmidt's lab. Eric Schmidt is a chemist. He purified, he looked for took this chlorine and dissolved it in methanol, purification, which is in the paper, and he found that if you look after HPLC purification, there was actually a band when you originally looked at it, and it's called islandamide, let's call it B, and let's see, paper, it's a polyketide, where is it? There it is. So it's a long, oh man, I can't draw that. Right, so it's this long polyketide. How can I draw bonds?
They look like big.
And there were two forms. So one is called the A form, and the other had actually one of these bonds was off. It's called the B form. And the people that originally characterized this, they characterized the A form. And what Chris found was, so A, when he did this scanning for the compound, he used UV, and the UV actually converted P to A. So what people had previously identified was actually the wrong compound. And so Chris just turned off the UV detector, and doing that, he was able to find that it was actually the B form. Okay. And it's this compound that he could purify, and I think we have a growth curve in there. Where you have salmonella cells, OD, over time, salmonella grows, but if you add the B, they die. So it really does look like a nice antibiotic. And then we can use simple genetics, which is to take our salmonella cells, right? Salmonella. Throw it on a plate, put it in the middle, you can put the B, and we got a nice zone of killing. No growth, killing. You wait another day. And we got mutants, right? Mutants that are resistant to killing. And so Chris then sequenced, did the genome sequence, and they're in a fatty acid synthesis gene called ACC, acyl-CoA carboxylase D protein. And you can see in the paper that there's an active site, so D actually interacts with another one, A, to make an active enzyme. And there's a place in this enzyme where the acyl-CoA binds. There's like a little tunnel, and he showed that the mutants were all in that tunnel where the islandamide was able to no longer allow this to bind. So that's where the inhibition was. So it binds in here, you get mutants in here that don't bind islandamide B, and now they can grow again. So we've identified a compound that inhibited salmonella, and using simple genetics, by looking for mutants that were resistant to the compound, he was able to show that the target was this acyl-CoA synthase. And that was great, but we did some tests on human cells. And guess what? Sadly, it also appears to inhibit the human acyl-CoA. But still, I mean, it's a nice exercise. So I would love to be able to do this in a way where we can, because there's hundreds, like a couple hundred Burkholderia species known, and they're all full of these gene islands, right, that are making secondary metabolites. Nobody knows what they are, and a lot of those could be new antibiotics, right? So we like to say, when we're talking about these people designing chemicals, raw material chemistry, or what Stan was talking about the other day, nothing beats 3 billion years of field testing. Nature has been building antibiotics for 3 billion years, right? So we should just go find them, not try to make them ourselves, right? So that's what we like to do. Anyway, that other thing that Chris had noticed, when he looked at the genome, he found there was actually 2 ACC genes in Burkholderia, right? And this one was in the same operon with the tylenamide genes. And the other one was somewhere else, suggesting that Burkholderia, very clever, it could make this antibiotic, but that antibiotic would inhibit its own ACC. So why not make its own ACC that was resistant to the antibiotic, right? And so Chris then took that, and you can see in Salmonella, he put it under Ravenous control, ACC1, ACC2, and he grew with a spot of the tylenamide, B, right? You saw he had here on one side of the plate, he had the ACC1 producing cell plus HERA, and he saw killing, but if he had the ACC2 producing Salmonella, he saw it was resistant. So that was pretty cool. That the bacteria in Halloween made an antibiotic, but it made its own enzyme resistant to its own antibiotic. So that was the story. That's how we got into this. Again, just a physician saying, hey, I have this idea, can we test it using genetics? And I said, sure, let's do it. Right? So I think it's a great way for antibiotic discovery. Again, it'd be great to have that T23 transposon, or whatever transposon you want. You can change trimethylprim resistance to whatever resistance that your particular bacteria can work with, right? And then ideally, you have a promoter out, and you can identify activators as well. Because you don't know necessarily what conditions are turning on the antibody, right? So that's why either you can knock out the repressor, and you don't have to worry about it, but also be nice to turn on any activator, so that you can control the conditions that turn on the production of the antibody. Okay? So that's our little story on antibiotic discovery and ventures into Burkholderia, which I never thought it ever worked. Okay. What else?
I think
Questions? Are we done? We're ready for lunch?
Alright.
we can always talk more if you feel like you're getting shortchanged.
So, questions?
Yes.
Paulina?
Yes.
So this doesn't happen in the wild type. The wild type strain
The wild type Burkholderia does not
doesn't
kill
produce.
salmonella on the plates because under a lab condition it's not turning on the antibody. Right? We have to force it using genetics
So the
to
thing is
knock out
that
the repressor.
this is very interesting because it was already repressed, no, in the wild type. So you needed to knock out that and still,
So presumably there's an environmental condition
yeah,
that causes the ATSR to come off and allow expression of the tylogamide synthesis genes, right? We don't know what that is. We don't have to know. We can use genetics. Right?
So like you, I know that the genome of Purcoveria has a lot of things there that we
Secondary
don't know.
metabolite stuff.
Yeah, so I had this idea of doing metabolomics because I wanted to know what compounds were producing.
But how are you going to know how to turn on those genes?
No, but just at different conditions.
Okay.
Just to know. But it didn't work because it's very difficult metabolomics.
Sure.
I mean, you need to know what part of the spectrum you have to check. So we only found carbohydrates or something like that.
Yeah.
So it didn't work.
That's why we're here, to tell you that genetics is easy.
I like it.
All you need are petri dishes.
You're bringing the word.
What's that?
You're bringing the word. You're the
Right.
prophet. You're the
Right.
prophet of
Right.
bacterial
This is the
genetics.
church of genetics. You're my sheep.
So I have another question. More questions?
Yes.
Yes.
I have two questions about the phage P22 system, and one is, what if DNA binding protein have 3 different binding sites, and I want to study the affinity? How can I apply it P22 technique, or there's another method for that? And the second one, what are the advances of phage P22?
Question at a time.
Oh, sorry.
No, you want to do all at once? We can answer one question, we can ask the next question.
Okay.
Otherwise, we can't remember. Stanley, you want to take this?
I didn't quite hear the question.
Okay.
So repeat the question, por favor.
The first question, what if a DNA binding protein have 3 different binding sites? I want to study the affinity. How can I apply P22 technique, or there's another method for that?
Yeah, so with this challenge phage technique, you can put each one of those sites onto the phage, and you can measure the relative affinity by the amount of the protein required to repress the phage. So the amount of IPTG that you add, you can titrate that and figure that out. Okay? So you can not only, once you've done that, you can not only figure the affinity for the different sites, but you can also then get mutations that affect those sites.
You could also look at the frequency of lysogeny with the same expression of your protein on the different binding sites in the challenge phage.
Yeah.
And at the higher affinity, there's going to be a lower frequency of, or a higher frequency of lysogeny because it's really shutting it off. So you can potentially see a gradation of the frequency of lysogeny depending on how well it binds.
But with the caveat that sometimes, even though in vivo you can see differences in affinity, this is a very sensitive system. So sometimes you have to turn down the plasmid rheostat. Does rheostat, everybody, rheostat translate?
I don't think so.
Expression.
Yeah, so rheostat's just a little device you turn and you get different
Oh,
amounts.
yes.
Turn up the volume.
Yeah.
Maybe the flag was the only one. Remember the word.
Question 2.
Question 2. The final question, what are the advances of phage P22 over other methods in terms of efficiency, specificity, and cost?
So if you're going to do a bunch of in vitro approaches, they're pretty expensive, right? They're expensive both because you need to have a variety of chemicals to do them, right? That cost money, but also it takes a lot of time to do them. In this case, the time is just building your initial system. Once you've built it by recombination, then testing the results are really straightforward. If you have something that you really want to get this done quickly, I would say send somebody to one of our labs and you spend 2 weeks in the lab, you're going to go home with answers and a kit to answer other things.
Yeah, as I mentioned, in my system, people looked at the binding, the major and minor groove affinity sites, but they couldn't tell, right? You could say it's binding both, but with this system, we could say the primary important binding is in the minor group, because if you knock that out, you get nothing. If you knock out the major group, you still get a good efficiency of binding. We could not see that any other way. So having an 8 order of magnitude range, it gives you huge sensitivity that you can't detect necessarily in vitro methods.
So if you're, let's just say you were doing a footprinting experiment. So, have any of you done footprinting, DNA footprinting? It is a pain in the rear. I mean, you really have to be very precise. But in footprinting, you could add different amounts of purified protein to the system. You got to purify the protein. In this system, you don't have to purify any protein. And in the footprinting experiment, you're doing it in vitro in a very artificial situation. In this case, you're doing it in the intracellular milieu, right? And in most cells, there are some commonalities about that. Relative amounts of magnesium being very similar, and so on and so forth.
That's the point I was going to make. So, you know, as a grad student, I worked with an enzyme, and it had mutants with different levels of activity in vivo, 20%, 50%, 80%, TS, not TS. In vitro, didn't correlate at all. So once you take them out of the natural system where they normally live and work, it can be completely different in vitro and not be meaningful. So if you do it in vivo, you know you're looking at the real system, right?
Thank you.
More questions?
Is it possible with this system to test if an RNA has a regulatory function?
Yeah, absolutely.
Yeah, you could put in so the ant mRNA, there's no reason in 5' UTR you can't put your favorite RNA binding site, RNA protein that binds the RNA. So you can look at regulation of translation, right? That's what you're getting at.
Yeah.
And that's, I've always trying to get a student to put remember that flag A that binds the RNA, I want to put that in here. That's that.
More questions? I have a, yeah, go ahead.
Do you have a question?
I have a question. Can we have the transposer?
Oh,
Or do I need to write to Theodora for that?
I'm sure I can just ask Colin if I can send it to you.
Okay.
Sure.
Let me give you a homework question.
Well, they've been very nice because honestly we haven't been discussing the papers. They're just telling the stories, which is nice. Because many of us don't understand the whole picture. For a few papers I didn't get the whole picture till you guys explain it. It's a relief that you guys are telling us the story.
And if you remember the story of Doug, geneticists are always nice and polite. Okay? Okay, so I talked to you the other day about chromosomes being very dynamic and changing. So within enteric bacteria, Salmonella, E. coli, the related ones, in those bacteria there are multiple copies of the rRNA operons. There's 7 copies. You look something like this. I get 7 right here. One, 3, 4, 5, 6, and 7. 7 copies. Our RNA operons encode the ribosomal RNAs. These are big sequences, very big sequences, right? And remember I told you earlier when I was talking about the rearrangements, and that even 10 base pairs is big enough for recombination to get rearrangements in the chromosome? These are really big, okay? KBs, right? So these potentially are hotspots for rearrangement. It's very interesting because if you look at Salmonella typhimurium, The organism that we know and love so well for its genetics abilities. And you compare it to a very close relative, Salmonella typhi. In Salmonella typhimurium, when you look at these, it's virtually always the same. It's just number them. You can get Salmonella typhimurium from somebody going to this bathroom here, some bathroom in Egypt, wherever it is, right? And you are always going to see this structure, right? In fact, that structure is important because people use this for mapping the chromosome to be able to distinguish strains. Now, if you look at Salmonella typhimurium, they scramble. Salmonella typhi. They're scrambled, right? So sometimes it will be 1, 3, 2, 5, 4, 6, and so on. Sometimes it will be anything else. Every combination you can think of. When you go to the outbreaks of typhoid fever, they're different. They're all different, right? What? 2 homework questions I want you to think about for next time. 1, why might this be uniform? When there's this much region that can undergo recombination, how come we're not seeing this? Right? Second one is, why is there so much recombination between the RRNs in type B? Okay? So you don't have to know the answer. I want you to think about why it might be. Right? This is really a thought question. Part A, Part B. Every good question has 5 parts, right? There's only 3 parts. Here's C. Okay? How could you design a genetic way to actually see movement between these bonds, to observe the recombination? In vivo. I told you that we see this happening a lot, but that's because we get a sample from one outbreak and we look at it, and then over here we get a sample from another one and we look at it, right? Now it's all typhi, but if we're just looking at the snapshot of one particular point in time, we want to see the movie of what, while it's really happening, so we can understand how it happens, right? So think about this, think about this, think about this. I'm not going to grade you on the answer, but I want to see a discussion about this. Okay?
Yes.
All right.
Okay, thank you, Stan. So, a couple of announcements. We're going to be able to make the picture in front of the mural. So, Martin went and talked to the people in the gate. Hopefully, there's not going to be any problem. If at any point they don't like it, we just don't argue and we just come back and see where we're going to get. That's the first one. So, we're moving over there in front of the mural. So, we're going to have the picture there. Martin is already there. Second, like I said, if there's any problem, let's just move to another place or leave it for tomorrow. We'll see. Okay? Because we don't want to get into any arguments. The second is once we're done with the picture, everybody's in the theory part are free to go. I will let you know which gate can you go, either the principal or come back all the way to the 5. So we don't know. Again, not complaining.
Oh, you know, when you see all these murals, it's always about these Mexican soldiers.
This one isn't doing this. So that's not very advanced. And the last announcement is for the laboratory. So after the picture, we'll take 20 minutes break, and then we'll come back to laboratory 4 to continue and finishing that part. I think we're finishing early. The laboratory today, we'll see. Okay? Thank you. See you tomorrow. Well, see you in a bit.